In the fight against Alzheimer's and Parkinson's, we have spent decades chasing what might be the wrong suspect. We've looked at the damage outside the neuron, assuming it was an attack from the outside in. But what if the destruction starts from within? Today, we are exploring a unifying theory of neurodegeneration, the convergent autophagic collapse. Let's start with the big picture. This is the funnel model. It proposes that neurodegenerative disease isn't just one thing going wrong, but a specific sequence of failures. Think of a funnel. At the top, you have a wide variety of causes: genetics, infections, toxins. But as you go down, they all get channeled into the exact same bottleneck. The theory here is that no matter how the problem starts, it ends in the same way: a catastrophic failure of the neuron's waste disposal system, known as the autophagy lysosomal pathway. To understand why this is revolutionary, we have to look at the prevailing view. For years, the amyloid cascade hypothesis has dominated the field. This is the outside-in view, the idea that sticky plaques accumulate outside the cell and kill it. But look at the bottom half of the screen. The convergent collapse theory flips this around to an inside-out framework. It argues that the neuron isn't a passive victim. Instead, the neuron's internal cleaning crew fails first. The debris we see outside, that's not the weapon, that's the wreckage left behind after the cell destroys itself. Let's travel down the funnel, starting at the wide rim, stage 1. This represents the diversity of insults our brains face. You see 3 distinct categories here: genetic factors, viral saboteurs, and environmental toxins. On the surface, a genetic mutation and a viral infection seem totally unrelated, but this model suggests they share a common ability. They all compromise the neuron's ability to clear waste. They are the initial triggers that push the cell toward the edge. Let's zoom in on the first input: genetics. We know certain mutations cause disease, but how? In Alzheimer's, PSEN1 impair the proton pumps that keep lysosomes acidic. In Parkinson's, LRRK2 mutations disrupt the traffic flow of vesicles. In ALS, C9orf72 expansions stop the cleaning process before it even starts. The takeaway here is that while the gene names differ, the functional outcome is the same. They all weaken the cell's degradative infrastructure. Next, we have a factor that is often overlooked: viruses. Viruses like herpes simplex, Coxsackie virus, and Zika are master saboteurs. They don't just infect a cell, they hijack its machinery. Look at the diagrams here. HSV1 literally binds to the start button of autophagy to stop the cell from eating the virus. Enteroviruses cut the fusion cables so trash bags can't reach the incinerator. They turn the cell's cleaning system into a construction site for more viruses, creating a massive backlog of waste. The third input is environmental and metabolic toxins. This is the wear and tear of life. Heavy metals like lead and cadmium can displace the magnesium needed to power cellular pumps. Then you have oxidative stress, the rust of the cellular world, which physically damages the machinery. And critically, look at the bottom right: toxic protein fragments like C99 in Alzheimer's. These aren't just bystanders, they act as an internal brake, jamming the gears of the acidification pumps. Now we move down into stage 2, the narrowing cone. This is where the magic of the theory happens. We've discussed 3 very different types of problems: genes, viruses, toxins. But notice how the funnel swirls them together. They don't cause random damage, they converge on a single choke point mechanism. They all attack the lysosome's ability to stay acidic. Without acid, the enzymes inside the lysosome can't digest anything. The recycling center shuts down. This implies there is a specific molecular nexus of failure. Meet the vATPase proton pump. This complex machine is responsible for pumping protons into the lysosome to keep the pH low. This diagram shows how everything we just discussed attacks this specific pump. Lead blocks its energy source, mutations prevent it from being assembled, oxidation warps its shape, and Alzheimer's fragments jam its motor. It is a perfect storm hitting one specific molecular machine. When this pump fails, the pH rises and the neuron is doomed. We are now at the neck of the funnel, stage 3. The pump has failed. The waste is piling up. The neuron enters a terminal state known as PANTHOS. The term comes from poisonous anthos, or poisonous flower, describing the shape the dying neuron takes. At this stage, the cell is locked in a futile cycle. It keeps trying to package waste into autophagomes, but it has nowhere to dump them. What does a PANTHOS neuron look like? It's terrifyingly distinct. Look at the blebbing on the surface. The cell is literally bulging with undigested trash. Inside, you see rosettes, or flower-like clusters of vacuoles. The dendrites and axons, the communication lines, become swollen and dystrophic. The neuron is essentially choking on its own waste. This state cannot last forever. We inevitably reach the final blow, lysosomal membrane permeability, or LMP. Think of this as the trash bag bursting inside the house. The lysosome is packed with toxic enzymes and iron. When its membrane finally ruptures from the pressure, those lethal contents spill into the cytoplasm. This triggers immediate, programmed committal death. The neuron doesn't just fade away, it collapses. Finally, we exit the funnel at stage 4. The neuron is dead, but something is left behind. According to the inside-out model, the amyloid plaque, that hallmark of Alzheimer's, is actually the remains of the fallen neuron. It is a tombstone. The dense core of the plaque is the undigested, protein-resistant debris that spilled out when the neuron exploded. History is fascinating because we often forget what we used to know. Back in 1907, Oscar Fischer described miliary necrosis, small spots of dead tissue. He saw the plaque as a site of death. Somewhere along the way, we forgot this and started blaming the plaque for causing the death. Modern research like that from Ralph Nixon's lab is bringing us back to Fischer's original insight. The plaque marks the spot where a neuron died of autophagic collapse. This changes everything about how we treat these diseases. If the plaque is just a tombstone, then attacking the plaque is like bulldozing a graveyard to cure a plague. It doesn't bring anyone back. The therapeutic window is before the collapse. We need to save the neuron while it's still struggling, not just clean up the mess after it's gone. It also explains why immune cells cluster around plaques. They are the undertakers trying to clean up the debris field. So let's recap the journey down the funnel. Stage 1, genes, viruses, and toxins stress the system. Stage 2, they converge to break the lysosomal acid pump. Stage 3, the neuron bloats with waste, entering the PANTHOS state. Stage 4, the cell ruptures, dies, and leaves behind a plaque as its tombstone. Where does this leave us? It leaves us with hope because we have new targets. Instead of just clearing plaques, we need to bolster cellular resilience. We need drugs that stabilize the vATPase pump and keep the lysosomes acidic. We need to activate genes like TFEB that build new lysosomes. And we need to detect this autophagic stress in patients years before the neurons actually die. The battle against neurodegeneration won't be won by fighting the ghosts of dead neurons. It will be won by empowering the living ones to clean their own house. Thanks for watching. If you found this breakdown of the convergent collapse theory helpful, make sure to subscribe for more deep dives into the mechanisms of biology.